Welcome back everyone to the 6th in my series of short talks on epigenetic science. In my last talk, we looked at the structure of the DNA double helix. What's truly amazing is how much DNA we have in almost every cell type in our body and how efficiently the cell is able to package such a large amount of DNA into a very small space. We're looking into how our DNA is condensed in the cell because it's a very important factor in our epigenetics. Here is an example of a human cell, the basic unit of our tissues, from the Your Genome site. Most cells aren't round, except some kinds of our blood cells, but it gives us a nice representation of the components in the cell. As we enter the cell, there are many parts called organelles. What's incredible is that our cells have the same systems as our body, a nervous system, a digestive system, a musculoskeletal system, etc. And our DNA is housed in a structure called the nucleus. The nucleus is about 6 micrometers in diameter, which is several times smaller than the thickness of a human hair. In fact, 10,000 nuclei could fit on the head of a sewing needle. Let's take a journey inside the nucleus. Now we're going through a nuclear pore inside the nucleus. Here we find the DNA tightly packed into what are called chromosomes. As humans, we have 23 pairs of chromosomes, one set from each parent, giving us a total of 46 chromosomes. What's kind of mind-blowing is that every cell has 2 meters of DNA if stretched end to end. A king-size bed is 2 meters wide, just to give us a frame of reference. By analogy, if a tennis ball represented the nucleus, you would have to pack in 24 miles of fine thread to account for all of the DNA. Wow, that's an incredibly large amount of DNA in every cell. We're going to go deeper into how so much DNA can fit into the nucleus because some of the epigenetic mechanisms are found in how our DNA is condensed. And as we start to go over more of the ways gene expression is controlled, this will empower us to apply epigenetics at a deeper level. My name is Robert Needleman. I'm a molecular biologist and biochemist. Thank you for joining me. Welcome to Empower My DNA. Our DNA does not exist alone. A chromosome is made up of DNA and proteins and another molecule called RNA. In fact, there is twice as much protein as DNA in every chromosome. A chromosome is one long, continuous strand of DNA. There are several stages of packing of the DNA. The first is called a nucleosome, where the DNA wraps around a set of 8 proteins called histones. Many nucleosomes are then packed together to form what is called chromatin. If we continue to unpack a chromosome, we would arrive at the DNA double helix. This is where we would see what is called a gene. We need to also expand our definition of a gene now. A gene codes for proteins or various types of RNA, which we'll cover in other talks. Here is an animation of how DNA is packaged into chromosomes from the DNA Learning Center. This is the DNA double helix at a magnification of 100 million times. You see the molecules vibrating in these animations because in cells, molecules are in constant motion. The purple molecules are the histone proteins, and you'll see the DNA wrapping around them. Once the DNA does 1.65 turns around the 8 histones, or histone octamer, a nucleosome is formed. Now we see many nucleosomes packing together. The magnification is less now because the chromatin we see being formed is a larger structure. The particular type of chromatin fiber here is 30 nanometers in diameter. Now the chromatin is packing even tighter together with other proteins that aren't shown here, and the smaller magnification tells us it's getting even bigger. You can see the chromatin wrapped into an even larger fiber here and eventually packaged further into a chromosome. Now you can see a cell with densely packed chromosomes at a magnification of only 3,000 times, which can be achieved with a light microscope. This is when a cell is preparing to divide, so the DNA is very condensed. Here you can see the DNA quickly condensing as the cell prepares to divide. The DNA is only packed into the most dense form of a chromosome for cell division. If you've learned about the DNA in our cells, you most likely were taught that DNA exists as condensed chromosomes, but cell division is only one phase of what's called the cell cycle. Most of the time, the cells aren't dividing, and the DNA chromosomes exist in what are called chromosome territories in the nucleus. Each one of these colors is a chromosome. It's really quite beautiful. This is from the Cavalli Lab at the Institute of Human Genetics. Chromatin is not randomly located, it's actually divided into compartments. This creates neighborhoods of gene expression. Here we see compartment A in red, which contains mainly active genes, and compartment B in blue, which has mainly inactive genes. In order for a gene to be expressed, it has to be made physically accessible from the proteins that are interwoven with the DNA. This is one form of epigenetic regulation. Here's an analogy where the DVD represents our DNA. DVDs that are in a case or sleeves cannot be read, just like genes that are encased in proteins cannot be read. The DVD has to be taken out of the case to be put into the player and read. The entire process of uncasing and reading the DVD was controlled and carried out by external factors, not the DVD. The same is true for our DNA. Here is an animation from Northwestern University created by Nicole Ethen. We can see the tightly packed chromatin. And as we start moving along the DNA, we see DNA winding around histone proteins forming many nucleosomes. The DNA is shown here in white, and the histone proteins are peach colored. Now we can see some of the epigenetic machinery, which are enzymes, approaching the nucleosome. A mark is being added by the enzyme to the histone tail, which causes a change in shape of the protein. The mark or tag is represented by the green light. You can see this allows the DNA to move more freely. Here we see an epigenetic mark being added to a histone protein, which frees the DNA and allows the protein machinery that reads genes to physically access a gene. Now we see this enzyme called RNA polymerase approach the DNA and express the gene. Here the gene is being read. After the gene is expressed, the DNA wraps back into a nucleosome. Placing epigenetic marks on the histones can either increase gene expression, as shown by the green marks and the DNA becoming more accessible, or it can decrease gene expression, as shown by the red marks and the DNA becoming less accessible and more compact. It all depends upon where the marks are placed. Here we see epigenetic machinery adding marks directly to the DNA. These are red in color because they will stop a gene from being expressed. The process of attaching and removing epigenetic tags or marks is dynamic, allowing cells to respond to various signals and regulate gene expression as needed. I hope these incredible animations are helping you get a glimpse into the powerful world of epigenetics. There are many forms of epigenetic control of gene expression. The ones I just showed you are by adding epigenetic tags or marks. Learning about how epigenetics actually works deepens our belief when we decide to affect our own gene expression. We can say things like, I focus on my body expressing genes for my health. I truly hope you're finding these talks helpful in your journey to becoming more empowered in your life. We have a lot more to explore in my next talks. Thank you so much for joining me. Take care and have a wonderful day.